Hi guys. This is my presentation of Chapter 16, How Genes Work. So Chapter 16 is a lot about gene expression. Gene expression is how the genes that you have are used to create your characteristics or to create the products that they make, mostly proteins. A couple of new terms for us are genotype and phenotype. Your genotype is what genes you have, or what information is in your genes. Your phenotype is your physical characteristics. So for instance, you could have a gene for blue eyes from one parent and a gene for brown eyes from your other parent. But you're only going to have either blue eyes or brown eyes, of course not both. So your actual eye color is your phenotype, then your genotype would be blue and brown eyes, and your phenotype would be one or the other. Every gene codes for something, some molecule. Almost all of your genes code for proteins, which means they have the instructions needed to build the proteins, but some of them code for RNA instead. Different types of RNA, like rRNA or tRNA. Then your phenotype is determined by two things. First, it's determined by your genotype and how those genes are used, so how the genes are expressed. And second, it's determined by your environment. So as an example, you might have two tall parents, so you would have genes for being tall genes. So your genotype would be tall. But if you were raised in an environment without enough nutrition, then you are not going to be able to reach your full height. So you would grow up to be short. You would have a short phenotype, and that's because of your environment, not because of your genes. In the next couple of chapters, we're going to be talking about how those genes are used to create your phenotype and how they're expressed. And the central dogma is all about how genes are expressed, so how they're actually used to make some type of product, usually a protein. So here you have a summary of the central dogma. You start with information in DNA that is put into mRNA in the process of transcription, and then the mRNA is used to make protein in the process called translation. So information starts in DNA, it goes into mRNA, and then it goes into protein. So this is the central dogma. This is the basic scheme of gene expression, using the genes in your DNA to make a protein product. Then kind of what proteins you have and your environment would be determining what your phenotype is, or what your actual physical characteristics are. This is a more detailed diagram where you're starting with again DNA and your genes at the top. So that is your genotype, what information you have in your genes. Those genes are used to make mRNA in transcription. Then the mRNA is used to make protein in translation, which involves a ribosome reading the mRNA and adding amino acids to the growing protein. Once that amino acid chain folds up, it'll be a complete protein. So that gene is now expressed, and then that protein is going to create some trait, such as your height or your eye color or your hair color, and that trait is your actual phenotype. And then you also have places where the environment can influence that process. So the environment is able to influence how mRNA is made from your genes, and it's able to influence how proteins are made from your mRNA. We're not going to really get into the details of how the environment affects it, but the environment can affect it at multiple points. So a little bit more on the central dogma. The central dogma is basically about information flow. So you have, where's my pointer? You have information in your DNA that then is put into your mRNA that is then put into protein. Transcription is the process of making mRNA from DNA, and then translation is the process of making protein from mRNA. And those two processes make up the central dogma, and they make up gene expression. For transcription, that relies on the enzyme RNA polymerase. RNA polymerase is the enzyme that actually makes mRNA based on DNA. Then translation relies on the ribosome, which is, as you know, the protein complex that actually makes the protein based on the mRNA. And of course, the ribosome has rRNA within it and also uses tRNA. The importance of tRNA we mentioned in lab, but we're going to talk more about it in the next chapter, actually. And just remember that this first process, transcription, making mRNA from DNA, that occurs in the nucleus of the cell. Then the mRNA has to be transported out of the nucleus to the cytoplasm to be translated. So translation happens in the cytoplasm and transcription happens in the nucleus. This is an example of gene expression and how that creates, or how the genotype creates your phenotype, or the phenotype of these little mice, that is. So here we have on the left a brown mouse who lives in the woods. He is brown to blend in with his environment. And then on the right we have a beach mouse who's white or pale in color. He lives on the beach and he blends in with his environment. So these two mice have a different phenotype. One is brown and one is, well, I guess not necessarily white, but white to tan, light tan. And these two different phenotypes are created by the different genotypes of the mice. So this wood mouse has, well, I guess first let me talk about how color is created in these mice in the first place. So melanin is the protein that creates color in your skin. It also creates the color in the mouse's coat. So all these cells that make up their little hairs are filled with melanin to give them the color. The wood mouse has a lot of melanin in his fur that makes him dark. The beach mouse only has a little bit of melanin that makes him light. Melanin is actually synthesized and put into the fur in response to a hormone called melanocortin hormone. In order for the cells in the mouse's skin to respond to that hormone, they have to have a receptor for it. Any cell, if it's going to respond to a hormone, has to have a receptor for that hormone. The receptor is a protein that is going to bind the hormone and cause something to happen inside of the cell. So when melanocortin hormone is released in both of these mice, it's going to go to the skin cells and bind this protein called the melanocortin receptor that is in the membrane of the skin cells. Once that receptor is bound to the hormone, that's going to kick off a series of changes inside the cell that results in melanin being made and put into the fur. So both of these mice have melanocortin hormone, and both of them have the melanocortin receptor. Both of them have the ability to make melanin and put it into their fur, but their melanocortin receptors are not the same. This mouse has one version of the receptor, or the wood mouse has one version of the receptor, and the beach mouse has a different version of that receptor. Here you have the actual protein sequence of the receptor. Of course, the receptor is very large. This is only 3 amino acids in that protein, but your wood mouse has an arginine. This is asparagine, and this is leucine. Those are 3 of the amino acids in this melanocortin receptor protein in the wood mouse. In the beach mouse, it has cysteine, then asparagine, and then leucine in its melanocortin receptor. So you have one amino acid that is different in the wood mouse's melanocortin receptor versus in the beach mouse's melanocortin receptor. That amino acid is different because the DNA is different for the gene for the melanocortin receptor in each mouse. For your wood mouse, it has a G, a guanine in the first place in this little snippet of the genetic sequence. For the beach mouse, it has an A there instead of a G. So that means that your beach mouse has a mutation in its melanocortin receptor gene that changed this G to an A, and the result of that is that you get, instead of an arginine, you get a cysteine, and that changes the function of the melanocortin receptor. So as first I should mention that Both of these genes in the wood mouse and in the beech mouse are the gene for the melanocortin receptor, but of course it's different versions of the gene. So we have a special name for that in biology. We call those different alleles. An allele is a version of the gene. So for instance, you have a gene for eye color. You can have brown eyes, green eyes, blue eyes, black eyes. So you would have different genes for all of those. Or sorry, not different genes, but different alleles. You would have a blue allele or a green allele, for instance, for your eye color. So for this wood mouse, he has one allele for his melanocortin receptor. That's one version of, that's going to make one version of the receptor. And the beech mouse has a different allele for the melanocortin receptor. That's going to make a different version of the receptor. The end result of having this mutation in the new allele that makes a cysteine instead of an arginine is that the melanocortin receptor doesn't work as well. All right, so this wood mouse with his arginine in the melanocortin receptor, that makes a receptor that works really well. So when his body releases the melanocortin hormone, it binds to the receptor, and the receptor makes his cells start making a lot of melanin and putting it into his fur, so he gets nice and dark. For this beech mouse, his melanocortin receptor doesn't work very well with this cysteine here, so when his body makes the melanocortin hormone, it goes to bind the melanocortin receptor, but it can't bind it very well. So his body doesn't make very much melanin and put it in his fur, and he ends up being pale. I'll point out his melanocortin receptor doesn't work well, it doesn't work good, but that doesn't mean that it's bad for him. For him, it's actually really good to have a receptor there that doesn't work well, because that means he ends up pale and blends in with his environment nicely. So for him, that's a useful adaptation. Right, so the phenotype of the wood mouse is brown. The phenotype of the beech mouse is light. The genotype of the wood mouse is one allele for this melanocortin receptor that has a G here. The phenotype for the beech mouse is a different allele for that same melanocortin receptor that has an A here instead of a G. And then this process of gene expression results in different versions of the melanocortin receptor protein in each mouse. Right, so the actual genetic sequence is the genotype. The actual color of the mouse is the phenotype, and taking the genetic information, putting it into mRNA, and then into the protein itself, that is gene expression by the central dogma. Okay, so this is the genetic code. You do not need to remember the genetic code. You do need to remember what types of codons you have and the characteristics of the code. So every three bases in the DNA makes up a codon. A codon is like a word in the genetic code. So if you have UUU, 3 U's in a row in your mRNA, that's one codon. That's one word of the genetic code, and it means something. In this case, it means phenylalanine, the amino acid phenylalanine. So each of these codons has one amino acid that it codes for most of them. Almost all of the codons code for an amino acid, so they are words that mean that amino acid. But you also have a couple exceptions. You have one start codon, that's AUG, that codes for methionine and for start. Methionine is an amino acid. Since that's also the start codon, every single protein always starts with a methionine. And I should mention this table here is in terms of the mRNA, which is why it has U's, but you could also make a similar table for the actual DNA that would have T's instead of U's. In addition to the start codon, you have stop codons. You have 3 different stop codons. Here's UAA, UAG, and then the third one is UGA. Those codons do not mean an amino acid, they mean stop instead. So when the ribosome is reading mRNA, it's going to look for the AUG, the start codon, and it's going to start reading there. it'll put a methionine, and then it'll go 3 bases down to the next codon and read that, and put whatever amino acid is coded for by that codon, and it will go until it gets to a stop codon. When it reaches the stop codon, it's going to stop and fall off of the mRNA, and the protein will be done, except that it will still need to fold. There are 5 characteristics of the genetic code. First, it is unambiguous. That means that each codon always means the same thing. So AUG always means methionine. It never means leucine or arginine or any other amino acid. It only means that one. So every codon always means the same thing. That is unambiguity. The genetic code is also non-overlapping, which means that you always read it by moving down 3 bases or 1 codon at a time. So as an example, if you have this stretch of bases in your mRNA, UGG, UCG, UAA, you are always going to read that as UGG, then UCG, then UAA. If it was overlapping after you read the UGG, maybe you would back up and read those 2 Gs again and get a GGU, and then back up again and read the GU again to get a GUC, something like that. This does not happen. It is non-overlapping, which means you always move down 3 bases to read the next codon. Another characteristic is that it is universal, almost. That means that every organism on the planet uses the same genetic code, with just a couple of small exceptions. For instance, in bacteria, they don't actually use methionine, they use a modified methionine. It's almost just like methionine, but it's a teensy bit different. So just with really small exceptions, every organism on Earth uses this genetic code. That, by the way, is some really powerful evidence that all life is related by evolution, or has evolved or has descended from a common ancestor by evolution. There we go. All right. If organisms had different sort of origin stories, if they came from different places, then you would expect that they would have a different genetic code. Since we all use the same genetic code, we all come from the same place. Next, the genetic code is redundant. Sometimes that's called degenerate instead. So sometimes it's called a redundant code, and sometimes it's called a degenerate code. The redundancy means that every amino acid usually has more than 1 codon that codes for it. So for instance, valine here has 4 different codons that all mean valine. So if you have a GUU, you're going to put a valine. If you have a GUA, the ribosome will put a valine. If you look at the table here, you'll notice that every amino acid except for methionine has multiple codons that code for it. Usually it's 4 or at least 2. Right, so for any given codon, it always codes for just 1 amino acid. That is how the code is unambiguous. But for any amino acid, it is coded for by multiple different codons. That's how the code is redundant or degenerate. And finally, the genetic code is conservative, which means that for all of these codons that code for the same amino acid, they always start with the same two bases, and it's the 3rd base that differs. So it's conserving the first 2 bases and only changing the 3rd base. And you can notice that pattern over and over again in this table, where all the codons that code for 1 amino acid share the same 2 bases and then differ in the 3rd base only. So the redundancy characteristics of the code make it less likely that if you have mutations, you're going to get a change in your protein. The conservative characteristic also helps reduce the effect of mutations, and we'll talk about how that works when we talk about the mutations, which we're going to talk about right now, actually. Mutations. So mutations are just changes in the genetic sequence. They're changes in what bases are where, or in the sequence of bases. When you have a mutation in a gene, that means that you now have a different version of that gene, which means that you have a different allele of the gene now, right? And because an allele is the fancy name for a different version of the gene. So by mutation, that's the only way that you can get new alleles or new versions of the gene. That means that you need mutation in order to have evolution, in order for genes to change over time, and in order for organisms to change, not individual organisms, but for populations to change over time. You have to have new alleles introduced. The only way to get that is by mutation. You have two very basic types of mutation, point mutation and chromosome mutations. Point mutation involves changing one single base. Chromosome mutation involves changing large stretches of DNA. You also have subtypes for each of those, and we're going to go into all the different subtypes. Before we talk about the types of mutations, though, we'll talk about the reading frame. The reading frame is just how the ribosome is dividing the mRNA up into codons. So here you have a stretch of mRNA. It's actually the same in each of these three panels. The sequence of bases is the same in each panel. And you're going to have a ribosome attaching to this mRNA and reading it to make protein. But there's different places where the ribosome could start. I mean, of course, in real life, it would find a start codon and start there, but for the purpose of this example, if our ribosome started here at the very beginning, it would read the first three bases as UCA, then it would go to the next three and read UGA, and go to the next three and read UCU. So it would be chunking the mRNA into codons in that way. On the other hand, in frame 2, your ribosome did not start at the very beginning. It started one base in. So that is chunking the bases into codons as CAU, and then GAU, and then CUC. So it's taking the exact same sequence of mRNA and reading it in different ways to get different codons. At the bottom, you have a third way that the ribosome could read this. It could start two bases in instead of one base in, and then it would read the first three bases as the codon AUG, the actual start codon, and then it would go to the next three and read AUC, then the next three and read UCG. So all these are different codons that give different amino acids, and therefore different proteins. And these ways, the ways of reading the mRNA that are different from between each of these three frames, those are the different reading frames. So depending on where you start, that is determining the reading frame. You're always going to be going from your starting point, three bases down, that's your first codon, so on and so forth. So depending on where you start, you're going to get different codons. Each starting point represents a different reading frame. There's one type of mutation that will come into play with. Okay, so here you have the types of point mutation, which again are mutations that affect only one base. On the top in this table, you have an original sequence of DNA in orange, then the original mRNA that's made from it below, then the original protein that's made from that. And then beneath you have the different mutation types, the different point mutation types, and how those affect the genetic sequence and then the ultimate protein as well. So first we have missense mutations. In a missense mutation, you are changing one base for another. So in this case, you start with a TGG as your second kind of codon in the DNA. And after your missense mutation, that last G has changed to a T. So you have TGT now. That results in a different codon in your mRNA. Then the ribosome actually puts a different amino acid there. So instead of a TRP, which is a tryptophan, it puts a cysteine instead. So In a missense mutation, it can have different final effects on the actual protein, on the amino acid sequence of the protein, but when you look at the genotype, at the genetic sequence, it always involves one base changing to a different base. Next you have silent mutations. In a silent mutation, you have a change in the genetic code. Sorry, a change in the genetic sequence, but it doesn't result in a change in the protein. So here you start with a TAT as your first codon in the DNA, in your original DNA. Then after the silent mutation, the last T has changed to a C. So now you have TAC. That gives you a different codon in the mRNA, but actually both of those codons, the UAC after the silent mutation and the UAU that you started with, both of those code for the same amino acid tyrosine. So you don't actually have a change in your protein. That's why it's called a silent mutation. Silent mutations are possible because the genetic code is redundant and conservative. So because you have multiple codons like UAU and UAC that all code for the same amino acid, it's possible to change one codon to another and still get the same amino acid. Because the genetic code is conservative, silent mutations usually involve the third base in a codon. So if you had changed the A here or the first T, that would give you a different codon. It's only the third codon that changes between, or sorry, the third base that changes between codons that code for the same amino acid. So if you change that base, you might get a codon that codes for the same acid. If you changed the first one, that would give you a codon that codes for something completely different. That would be a missense mutation, not a silent mutation. Next, you can have a nonsense mutation. In a nonsense mutation, one base changes to a different base, and that changes whichever codon was there originally to a stop codon. So it introduces a stop codon. So let's see, in our original genetic sequence, we had a TGG as our second codon. That results in a UGG mRNA codon. After our nonsense mutation, we've changed that second G to an A, so that now in our mRNA we get UGA, and that's one of the stop codons. So when your ribosome is reading this mRNA, it's going to get to the mutated UGA and it's just going to stop. That gives you a protein that is too short. Usually that means that it doesn't function since it's too short. Next and last, you have a frameshift mutation, which is the most serious type of point mutation in a lot of ways. In a frameshift mutation, you're not changing a base to a different base. Instead, you are adding a base or deleting a base entirely, and that changes the reading frame. So in this case, we started with TGG. In our frameshift mutation, here's our T and the G and the last G, but we added a C. So now we have TCG instead of TGG. If you look at the next codon, we started with CTA, now we have GCT. If you look at the next codon, instead of GTA, now we have AGT. If you look at the last codon, instead of CAT, now we have ACA. And then we have one extra base. So every single amino acid from this point on, from where you get the mutation on, is probably going to be different. So you're going to make a protein that is completely different from the one that you were originally making using that gene. So out of these types of mutations, the frameshift mutations are virtually always bad, real bad. Usually you get a protein that doesn't work at all. The nonsense mutations are the same thing. They're usually bad, real bad, because you're getting a protein that's too short. The exception would be if you had a nonsense mutation that was at the end of the gene anyways, then you would get a protein that's only a little bit too short, so that might still function fine. Your silent mutations don't have any effect at all. They're neutral, since the protein you make is still the same in a silent mutation. Your missense mutations can be good, they can be neutral, or they could be bad. Usually they're bad, sometimes they're neutral, and occasionally they're good. Occasionally you get a protein that works a little better than it used to, or has a new function that it didn't used to have. So here's an example of how those different point mutation types affect the genetic sequence, or the genetic, how the ribosome is reading the genetic sequence using a sentence, right? So every codon is 3 bases, and it's like a word and then genetic code. So here we have a sentence made out of 3-letter words. So first, our original sentence is the fat cat ate the rat. If you have a missense mutation, that means you're going to change one letter to a different letter, giving you a different word. So you might change your sentence to the fat cat ate the rat. The sentence still mostly makes sense, but it doesn't entirely make sense. If you had a silent mutation, you would change one letter, but that would not change the word. So you would still have the fat cat ate the rat. If you had a nonsense mutation, you would introduce a stop codon, so the sentence would end up too short. So you would end up with just maybe the fat. And if you had a frameshift mutation, you're going to add a letter or delete a letter entirely, and that's going to have a drastic effect on the sentence. So you might get the tet, her, at, and it makes no sense at all. Okay, these are the types of chromosome mutations, which are mutations involving large stretches of DNA, either a big part of a chromosome or sometimes an entire chromosome. And remember that a chromosome is a large molecule of DNA. So first, sometimes when cells divide, things go wrong and you end up with changes in the number of chromosome. Usually that's due to non-disjunction, which is illustrated here. So we didn't really go over mitosis and meiosis or how cells divide yet. We will in the next couple of weeks, but when a cell is going to divide, it prepares for that by copying all of its DNA, so it ends up with extra copies or another copy of every chromosome. And then those are all going to line up in the middle of the cell. So all these 3 X's would be the copied chromosomes. So 1/2 of each of those X's is a full chromosome, actually. You would actually have started out with little lines like this, and then when you, that's one chromosome, and when you copy it, you end up with this little X. So those are supposed to line up, and then they're supposed to split apart so that 1/2 goes to each side of the cell. Then the cell kind of splits down the middle, so you end up with 2 new cells that are each supposed to have 1/2 or one copy of each of those chromosomes. Sometimes the chromosomes don't split though. So this guy at the top, he has not lined up correctly, and he is not going to split correctly. That is non-disjunction. So when all these other chromosomes split, so that each copy goes to one side of the opposite sides of the cell, this guy didn't split. The entirety of him is going to just this one side of the cell. We call him one chromosome because he's one molecule, since he's attached to himself in the middle, but he actually has 2 copies of one chromosome within him. So that this cell, this daughter cell, is missing that chromosome entirely, and this daughter cell has 2 copies of the same chromosome. So that's a non-disjunction event. That is a mutation that causes a difference in the number of chromosomes in different cells. Usually if that happens to an animal cell, the cell is just going to die. If that happens in a plant, though, then the cell may be able to live, and that'll just be a new mutation that's been introduced. Apart from changes in the number of chromosomes, you could have changes to the chromosomes themselves. There's 4 types of chromosome mutations that are like that. First, you have deletion, in a deletion mutation, oh boy, where's my pointer? There we go. In a deletion mutation, you cut out part of the chromosome. So here, this part that has gene A and gene B is cut out entirely. You end up with a shorter chromosome that's missing some of the genes that were on it. You could also have an inversion mutation, where you take a segment of the chromosome, cut it out, flip it around the other way, and then put it back in. So instead of having first gene A, then gene B, you end up with first gene B, and then gene A. Then you could have a duplication mutation where part of a chromosome is copied and you basically end up having it twice. So here in your original chromosome you have gene A, then gene B, and your duplicated, or in your new mutated chromosome that had a duplication mutation, you have A, then gene B, then another gene A, and another gene B. This can actually happen again. It can happen multiple times. You end up with many, many copies of the same gene. Then each of those genes can start accumulating point mutations and turn or evolve basically into different genes. That gives you what we call gene families. So you have a lot of genes that are all related to each other, and they're all part of the same gene family. And the way that they got there is by these duplication mutations. So let's say that you need gene A for something that's real critical, so nothing can go wrong with gene A. If you have a mutation in gene A, you just die. After a duplication mutation, you have two gene A's. So as long as this gene A doesn't mutate, you're going to be fine. Then this gene A can start mutating however random chance causes it to mutate, and you'll still have your critical function served by the first gene A. So then the second gene A can mutate freely and can take on new functions. And then you have the beginnings of a gene family. Finally, you can have translocation mutations. Those involve two chromosomes exchanging pieces of each other, of themselves with each other. So here in our first chromosome, in our big first chromosome, we have gene B and then gene C. And then we have another original chromosome, which is little, that has gene D and gene E, and we're just going to switch out these segments so that now your big chromosome has gene D and E, and your little chromosome got gene B and C. So a lot of the time, those mutations are bad. In particular, a deletion mutation where you're just losing genes, that's almost always bad, real bad. Some of the other mutations like the inversion mutation and the translocation mutation, those could be kind of neutral. They might not have a big effect, but they might also be bad because since the genes are ending up in a new place, they might end up being regulated differently or expressed differently. I guess when we talk in the next chapter about transcription, we're going to talk a little bit about gene regulation. That is how the cell is controlling which genes are used to make proteins at any given time. If you move a gene to a new place, you might start regulating it differently so that you use it to make protein at different times, or maybe too much, or maybe not enough. That could lead to problems with the functioning of the cell. That can also lead to cancer. Duplication mutations frequently are not bad. They actually can be good because they can give rise to gene families. So you can get extra copies of the genes that can take on different functions. So you gain new functions for the cell. That's a good thing. But sometimes you might end up with too much of those proteins. So let's say that gene A over here, it got duplicated, so now you have two of it, and it does not mutate right away, but you do right away start making double the amount of this protein. That might be bad for you. That might end up being toxic for the cell. Chromosome mutations are frequently associated with cancer. If you look at the genotypes of cancer cells, a lot of the time you'll see chromosome mutations where they're missing chunks of chromosomes, or they've got extra chunks added to their chromosomes, or the chunks are moved around to different places on different chromosomes. It's real common to find chromosome mutations in cancer cells, and is probably related to the development of cancer in the first place. In order for a cell to avoid becoming cancerous, it needs to have control of its gene expression, which means it needs to have gene regulation. When you have these chromosome mutations, a lot of the time that can disrupt your gene regulation, so that you start using genes you're not supposed to use, or using them too much or too little. That can lead to cancer. Finally, we can categorize mutations by the actual event that happens, which we just went through, or we could categorize them by the effect that they have. They can either be beneficial or neutral or deleterious. Beneficial means good, neutral means no effect, of course, and deleterious means bad. On the test, I'll just say good and bad, not beneficial and deleterious. In general, just as general rule, most mutations are bad for the individual. But of course, because we need mutations to get new alleles, mutations are good for kind of the species or life as a whole, because that gives us diversity. That's why we don't all look identical to each other, and actually that's why we're not all a single-celled organism still. But as far as the benefit for the individual, mutations could be good, neutral, or bad, and most of them are bad. For your chromosome mutations, the deletion ones are bad. The duplication ones could be good. The inversion and translocation ones are probably bad, could be neutral. The duplication one could also be neutral, or it could be bad, but it's more likely to be good. So I guess the duplication is really the only type of chromosome mutation that has that shot for being good. For your point mutations, silent mutations are always neutral by definition. Missense mutations are usually bad. They could be neutral, or they could some of the time be good, like a small percentage of the time. Rarely they can be good, adding new functions. Or even, I guess, sometimes deleting a function that you don't need anymore, like with that beach mouse. He had a missense mutation in his melanocortin receptor that made the receptor not work as good, but it ended up being a good thing for him when he was trying to live on the beach. Then you also have the nonsense and the frameshift mutations. Those are almost always bad. Except I will add that for the nonsense mutations, occasionally they're good. Sometimes they're neutral. If you got the nonsense mutation at the end of the protein, so it's only a teensy bit shorter, that probably would be neutral. And occasionally you end up with a protein that actually has a new function when it's shorter. That's not very, as I mentioned, that's not very common, but it can happen. And that's chapter 16.